Welcome to our focus topic screencast, which is on Chapter 9. And we want to know what the heck is an action potential. And I'm going to show you the grand scheme of how an action potential works within our muscular system. So the first thing I want to do is talk about a nerve stimulus at our neuromuscular junction. But before we get into all that, I just want to give you a general overview of our nervous system. When we attain some kind of stimulus, whether it's through sight or touch or taste or we hear something, all of that information is coming through a neuron. And that neuron is a sensory neuron. We're sensing something and sending that information into our body to reach our brain. So we're getting sensory input through that sensory neuron. Then we go to our integration center, which essentially is our brain, but sometimes we have reflexes in our spinal cord, but we're going to talk about all of those more detailed things in our nervous system lecture in Anatomy 3. So anyway, we get that information, that sensory input, and our brain determines what output we're going to do. So then we send information out a motor neuron, or we can call this, when we're our motor output affecting skeletal muscle, we can call this a somatic motor neuron. Now, soma means body, so really this is affecting our skeletal muscles that controls our body, or we could call this neuron the motor neuron of our somatic nervous system. And we know skeletal muscle is under voluntary control, so that means we have conscious control. We tell we're kind of telling our nerves when to send an impulse to our skeletal muscle and cause contraction. So now that you know all that, let's talk about our neuromuscular junction. So a motor nerve is going from my brain, information from my brain is moving down my spinal cord, out some spinal nerves, and let's just choose a specific one. We'll choose the musculocutaneous nerve, since you studied this nerve kind of in Chapter 10, because you know the musculocutaneous nerve innervates all of those anterior arm muscles, right? And so one of those anterior arm muscles pictured here is our biceps brachii. So this nerve is getting a nerve impulse, an action potential being sent through it. So we're getting this wave being sent through our musculocutaneous nerve, and it's going to reach our neuromuscular junction, or NMJ. So what our neuromuscular junction consists of, and I'm just going to go back to this image here. At the very end of our neuron, you see this is our axon. At the very end of our neuron, we have these terminals, and the terminals are almost like swellings. So that's what we're seeing in this image here. It's at the very end of that nerve we have these synaptic terminals, and at these synaptic terminals or axon terminals, we have a space between the actual nerve and our muscle, and that space is called the synaptic cleft, and we get a synapse that occurs within the synaptic cleft. What a synapse is the neurotransmitters, in this case acetylcholine, moving across that space to get to whatever that target organ is. In this case, it's our skeletal muscle. Now, our skeletal muscle, you could see this plasma membrane, which is called the sarcolemma. Remember, sarco meaning muscle, lemma means plasma membrane. So this is our muscular plasma membrane, has all of these folds within it. They're highly folded, and this is really great because it increases the surface area for millions of our acetylcholine receptors, okay, these little purple guys here. So what's going on here? We get an action potential sent through the nerve. Remember, action potential is the same thing as a nerve impulse, and that nerve impulse causes calcium to move into our synaptic or axon terminal. Now, when calcium moves on in, it tells acetylcholine to leave the neuron through exocytosis. Acetylcholine then moves through our synaptic cleft and attaches to these acetylcholine receptors on the motor end plate. Okay, motor end plate, again, highly folded for more surface area for our acetylcholine receptors. Okay, hold on, let's get to the right slide here. Now this is another view of our neuromuscular junction. So let's just review what we explained in the previous slide. So here at the number 1, we're seeing our axon terminal or synaptic terminal with our acetylcholine, and it's releasing it into the synaptic cleft to attach to our acetylcholine receptors on our motor end plate. Now we said when that happens, that binding is going to allow our sodium ions to enter the cell and our potassium ions to exit the cell. So we see that happening right here at this blue receptor. We're going to get more sodium moving in than potassium moving out. Now I want to talk about what a membrane potential is. Now membrane potential is going to be a difference in electric potential between the interior of the cell, which would be all of this area in here, interior of the cell, and the exterior of the cell. Typically this is going to range anywhere from negative millivolts to 90 millivolts. Millivolts are just a unit of electrical potential. So remember in our chapter 2 lecture in Anatomy 1, we were talking about when a molecule is polar and when it is not, and we used the example of water as being a polar molecule, and that was because oxygen sitting in the middle here had more of a negative electric charge to it, so it would hold the electrons closer to it, right? Because we have one electron from our hydrogen. Okay, and I'm just going to draw this in. Here's my other hydrogen, and it would share another electron from its other hydrogen, because in our valence shell, remember, oxygen has 6. Okay, let's draw this out again. There's my oxygen. I got 2 electrons in that first shell, and then I only have 6 in that valence shell. And what do we need in our valence shell to make it happy? We need to fulfill that octet rule, right? So we need 8 electrons here, and that's why it shares the other 2 electrons from the hydrogen, and then oxygen is happy. So it has its 8 electrons here. Okay, so 1, 2, 3. And so this becomes more negative because it wants to hold those electrons closer to itself, whereas hydrogens, they'll become more positive. So this is now polarized because we have more of a negative charge in one area and more of a positive charge in another, whereas something like carbon dioxide, that's sharing its electrons nice and evenly, so that's why that's nonpolar. Anyway, we can think of the plasma membrane as being polarized because the inside is more negative than the outside. Typically, skeletal muscle is going to have negative 90 millivolts as far as its membrane potential. Whoops, don't know why my M looks so funky, but negative 90 millivolts on the inside of the cell. So when we start to take in more sodium inside of the cell, that causes the inside of the cell to be more positive. We're going to start moving closer to a positive state inside the cell. So when we do that, that will propagate the action potential through the plasma membrane. Okay. So once we get this channel opening up and more sodium moving into the cell and more potassium moving out of the cell, we want to get rid of this acetylcholine as long as our nerve stimulation isn't there anymore. So the way we get rid of this is we use an enzyme called acetylcholinesterase. That's going to break acetylcholine up into acetic acid and choline and take it back to the neuron to be reused again as acetylcholine. Now let's take a closer look at what's going on with the channels in order to get the action potential sent through our muscle. And I just want to clarify, an action potential takes place in the nerve, so we get a nerve impulse being sent through, but we also get an impulse once this channel opens up and allows an influx of the sodium. We also get an action potential being sent through the plasma membrane of our muscle. So first thing that happens is we get a binding of acetylcholine to our receptor. That's opening up our chemically ligand-gated ion channels and allowing more sodium moving into the cell, and potassium moves out of the cell. Now that's because the driving force for our sodium moving into the cell is greater than our potassium moving out. So we get more sodium diffusing in than our potassium diffusing out. Now, we're at this point here. We're going to start to get more of a positive state inside the cell. So now we're going to have our sodium channels, the voltage gated ones, opening up. And what I mean by voltage gated channels, it means that when the voltage is changing inside the cell, becoming more positive, that's when those channels open up and let the sodium pour on into the cell. So sodium enters and we get more and more positive, and our membrane voltage threshold, which is really right around positive 30, is when the action potential is generated. So here is the positiveness on the inside of my cell. From -90, we're getting more and more positive, and we reach our threshold of positive 30. This peak here is where the action potential is taking place. Now, once the action potential happens, we are going to close our sodium channels and open up the potassium channels. But let's say the action potential, let me use a good color here. Let's say the action potential is taking place here. This is going to be our muscle cell. Okay, we get our positive 30 in this region of our sarcolemma. Then that's going to propagate down the rest of the sarcolemma and cause the adjacent area to start becoming more and more positive too. And the way it does that is we have these voltage gated channels for sodium that will also open up. So it's kind of like a cascade effect. Once this reaches its threshold of positive 30, then it's going to continue down the sarcolemma to cause the next part to move to positive 30. And so these action potentials are eventually going to cause the contraction, which we'll get to in the next slide. Now let's get back to what happens after the action potential and these sodium channels close. So now our potassium isn't allowed into the cell anymore, making it more positive, and we are going to have our potassium that is found inside of the cell. Okay, that's going to start flowing out. Okay, and when we get this potassium moving out of the cell, that's causing the cell to become more negative, and we move back down to our resting membrane potential, which is -90 millivolts. And at that point, potassium voltage gated channels are going to close. Okay, now I want to kind of put this all together for you, kind of do a summary and also put it together with our excitation contraction coupling, as well as the cross bridge that's going to occur between actin and myosin. So we have a nerve impulse or action potential sent through the nerve. These acetylcholine vesicles within the nerve are then going to release acetylcholine through the synaptic cleft and to the acetylcholine receptors on the motor end plate of the sarcolemma on our muscle cell. Now when that happens, our sodium-potassium channels are going to open up. We let more sodium into the cell, and that causes the inside of the cell to become more positive. When it becomes positive to the level of positive 30 millivolts, that threshold, then the action potential is going to be generated and starts to propagate down the sarcolemma. Now, when it does that, the action potential is going to move down these T-tubules. Now, T-tubules have a little receptor here, and when that action potential moves on through, that receptor is going to change shape and cause the other receptor on our sarcoplasmic reticulum to allow calcium to move out of the sarcoplasmic reticulum and onto our myofibril. And our myofibrils are going to be made up of a bunch of sarcomeres that contain our actin filaments and our myosin filaments. So I'm going to close up here to what's going on in this area. So let me repeat myself. Remember that action potential gets sent down the sarcolemma and triggers, moving down the T-tubule, triggers a change in this receptor, this protein here on our T-tubule, excuse me. And when it changes shape, that causes these channels, these calcium channels, to open on up, and the calcium moves from our sarcoplasmic reticulum, or the SR, into our myofibril. Now we could see a little bit better here in this zoomed in image, but let's go ahead and look over here where it's broken down even more. These calcium ions are then going to move onto our protein on actin, troponin. When calcium binds to troponin, that causes a shifting of our tropomyosin that covers those active sites on the actin filament to move out of the way. So once that tropomyosin moves out of the way, these active sites are now available for our myosin heads to attach. And remember, the cross bridge formation is going to start when we have an energized myosin head, okay? And we need our ADP and inorganic phosphate to leave the myosin head in order for the power stroke to occur. And that means we're shifting this actin filament in toward the M line. So here's our M line here in the background. So these actin filaments are going to start moving on in, and that is causing the muscle fiber to shorten. So I think that was my last slide. I hope this kind of clarifies a little bit of the material for you. If you still want me to re-explain any portion of it, please let me know, and I'd be happy to do so.